Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. Today is November 6, 2025, and we're talking to Dr. Jason O'Connor. Jason is Associate Professor of Pharmacology at the UT San Antonio Long School of Medicine and the Audie Murphy VA Hospital. So he's one of our own, and didn't have a long commute.
No, I did not.
And Jason's research is at the intersection of inflammation, depression, learning, cognition, aging, and metabolic networks. So, hi, Jason.
Hi, I'm glad to be here. Thanks for having me.
Thank you. Hey, I'm your host, Charlie Wilson. So I think everybody is familiar with the psychological effects of having an infectious disease. And just from my own experience, I would list them, the fatigue, sleepiness, trouble concentrating, desire to kind of isolate yourself, even light sensitivity, and avoiding challenging intellectual or interpersonal situations. And these symptoms seem to not matter too much about what's causing me to get sick. That happens when I have the flu or when I have a kidney infection. Mm-hmm. So I think it must be my immune system's response to the disease that's affecting my psychology.
Yeah.
And those symptoms are at least a little bit similar to what happens in depression.
Yeah.
So is there a connection?
Yeah. So I would say that assessment is pretty well spot on, right? And so the idea that as we talked about in the seminar earlier today with the students, that like the immune system kind of takes on this manifestation as a sixth sense, if you will, as a way for our body to respond to infection or tissue damage or things that really anything that is out of the ordinary, that's not supposed to be there. Our immune system's job is to respond and repair, like return us to health. And in doing so, the symptoms you listed or mentioned, we're all familiar with them on some level, like just the general being sick. But it was, I guess it's notable that these things are studied in a laboratory as well. So we do know, to your question that sounds like your immune system, and we do know the molecules that the immune system secretes in response to a pathogen cause those things to change. We can experimentally cause them to change in humans or in animals, and we can intervene in some of those symptoms if we take measures to block or inhibit specific molecules or pathways. So, and it is also true that those symptoms of depression or the behaviors and things that you list off, they do resemble, or symptoms of sickness, they do resemble symptoms that are described by individuals who are experiencing depression. And that's an observation that was really being made in the literature about the time that I was starting to think about transitioning from graduate school to a postdoc. And so I guess we're talking in the early 2000s. That the scientific community was really starting to wonder if the immune system and the molecules of the immune system can in fact change the function of our central nervous system, our perception of the world around us and how we behave and how we act. And those are symptoms experienced by people suffering from depression. Is there overlap? Can we learn something about depression from studying the mechanisms of sickness in a laboratory? And so our lab and many other labs have been working on this for the last 20 or 25 years, and we've come a long ways. And our lab in particular has identified some metabolic pathways. So you mentioned we're at the interface or intersection of the immune system and the brain and psychology, psychiatry, and metabolic pathways. The metabolic pathway that we're focused on in our lab has to do with the breakdown of tryptophan, which is an essential amino acid that we get in our diet. We think that is in part responsible for mediating some of the effects of the immune system that the immune system is having on the brain.
So we're all familiar with one of the metabolites of tryptophan, which is serotonin, and which we associate with depression. Yeah. But that's not the one, right?
That's not. So the pathway that I study is called the kynurenine pathway. And so you have to practice saying that on your own a few times.
I have been.
It's usually a little tricky for people. And the intermediate metabolites, the names get long, so we start to use acronyms to make it easier. But for sure, I like to say that when people think about tryptophan, they think about it in one of two ways. If they're a neuroscience-minded person or have some scientific background, they think about serotonin. Like tryptophan is the precursor for serotonin's biosynthesis. And everybody else thinks about tryptophan as what makes you sleepy after you eat Thanksgiving dinner, after your turkey dinner. And they are both true. And it is also true that very little tryptophan goes to either of those fates. So on balance, tryptophan being an essential amino acid means we have to get it from our diet. But where it goes is predominantly to kynurenine metabolism in the liver. So some tryptophan goes to build proteins, amino acids being the building blocks of proteins, but tryptophan is a low abundance amino acid in proteins. It has this big bulky side group that makes it folding difficult, and so it's not a predominant amino acid in proteins. A little bit goes to serotonin, a little bit goes to serotonin, but most 90% or so in a healthy, happy individual, adult individual, 90% or so is metabolized along this kynurenine pathway in the liver. And this is because it is broken down to generate a cofactor called NAD, which is important for energy biosynthesis or energy reactions within the cell.
But some of the intermediates along that route are familiar to neuroscientists as the NMDA receptor agonist or antagonist.
That's right. And so while I just said kynurenine happens all in the liver, that's not the whole story. So during conditions of inflammation or immune activation that can be triggered by any number of things, infection or injury or illness or disease, the enzyme machinery that metabolizes kynurenine to these intermediates that I'll get to in just a second, they increase outside the liver in immune cells in circulation, in immune cells that are resident to tissues like the brain and the kidney and adipose, and other cells as well. And when that happens, then we start to see changes in these tryptophan kynurenine metabolites in circulation and in tissues. And as you mentioned, two of the, I guess, better known metabolites are called quinolinic acid with a Q or kynurenic acid with a K. And they do have opposing effects on NMDA receptors, quinolinic acid being an agonist, kynurenic acid being an antagonist. But it's also true that several of the intermediate metabolites that are made en route to production of those are also neurochemically active or capable of changing redox states and potentiating oxidative stress and things that also are known to impact the function of the cells in the environment. So if this is happening in the brain, these things together, under the right circumstances, right conditions, could create an environment where the neurons or the circuits, the things that the structural organic elements that dictate how we sleep, how we eat, how we feel, those things may work less well when metabolites that aren't supposed to be there start showing up and making the system dysfunctional.
So the metabolites are all coming from the liver, or are some of them being generated locally in the tissues?
Both. So the majority of kynurenine So that first principal metabolite of tryptophan, the majority of kynurenine that gets into the brain comes straight from circulation, which may be the liver, but it may also be immune cells in circulation or other tissues. When it's in the brain, then it can be taken up and metabolized by the cells that are resident to the brain. And that includes neuronal and non-neuronal cells. Now, my lab tends to focus on a non-neuronal cell type called a microglia, which is oftentimes referred to as the resident innate immune cell of the brain. While it's an oversimplification, it's also true. And these cells, the microglia, are believed to disproportionately metabolize kynurenine towards quinolinic acid, the oxidative pathway that generates excitatory and more reactive intermediate metabolites. Whereas astrocytes resident to the brain, so these are another non-neuronal glial cell type that serve a support function to neurons and synapses, they metabolize kynurenine predominantly in a different direction. They transaminate it and create kynurenic acid. And what we've learned over the years is that manipulating this metabolic pathway can inhibit or potentiate or change the behavioral phenotypes that occur in response to inflammation in laboratory animals. So we've come to the conclusion, based on years of work in our lab and in others, that these metabolites at least play an important role. They may not be the end of the story or the whole story, but they play an important role likely in mediating the effects of inflammatory cytokines, inflammatory molecules on brain function.
So if you just give me some of those metabolites, will I experience those symptoms? And if so, do you have to put them into my brain, or?
That's a good question. So we've never injected them straight into a human being, but I could say we have done those experiments in the laboratory setting in animal models, and we are able to recreate or recapitulate some of the behavioral responses to immune activation without activating the immune system at all, but just by direct administration of kynurenine metabolites that are either fate committed. So that is to say that when that kynurenine is metabolized oxidatively, it forms a compound called 3-hydroxykynurenine. We can inject 3-hydroxykynurenine systemically into a mouse, and in a dose-dependent manner, it causes many, maybe not all, but many of the same behavioral responses that those mice will elicit in response to immune activation.
And it doesn't have to be placed into the brain.
It doesn't.
Anywhere.
No, so that's actually, so I didn't catch this, or I didn't comment on this initially when you asked that, but it's a good point. Some of these kynurenine metabolites don't cross the blood-brain barrier. So there's a special neurovasculature that some of the audience or you're well aware of, that the brain has a unique neurovasculature that is in large part less permissive to molecules crossing. So perhaps it serves to protect the brain or restrict bad things from getting into the brain. Some of these kynurenine metabolites are actively transported. For example, kynurenine itself shares the same transporter to cross from the periphery to the brain as tryptophan and the other large neutral amino acids that are structurally similar. So that's one mechanism. Some of the intermediate metabolites they're charged molecules, so they have a polarity to them that makes them
not dissolve in the membrane.
Not dissolve. They don't pass lipid membranes, so they are restricted to the periphery. Kynurenic acid and quinolinic acid are two of those. However, 3-hydroxykynurenine, the intermediate metabolite that I just mentioned injecting, that is hydrophobic, which means it will cross cell membranes. And so it will get from the periphery into the brain. There may be other mechanisms that we're not aware of, but we do know that when we inject it systemically, we see a rapid increase in the periphery, followed by a rapid increase in the brain.
So you can duplicate these symptoms with those metabolites. That shows they can do it, but can you do away with the symptoms by blocking them to show that that's the only way to do it?
Right. So, and that's how we approach these questions, right? So we make an initial observation that suggests that this pathway or these metabolites might be important during conditions of inflammation, but inflammation has a lot of moving parts. To isolate it down to one thing is tricky. So we inject a single metabolite. We recreate the profile, the behavioral profile. That gives us some additional confidence that maybe these metabolites are in fact causing the effect. Another way that we can approach that is to use genetically modified animals or transgenic animals that have a targeted deletion in key enzymes in this pathway. And so we've done that in some cases. And in one of those cases, we've generated a strain of mouse that does not express that enzyme that converts kynurenine to 3-hydroxykynurenine. So that's called kynurenine monooxygenase, or KMO. These KMO knockout mice do not express all of the behavioral responses to LPS that a wild-type mouse.
LPS is infectious.
I didn't. Yeah, so I've been saying immune activation. So LPS, or lipopolysaccharide, is one of the tools that we use to activate the immune response. Right. So if we activate the immune response in a wild-type animal, we see those symptom profile, the behavior profile that we were talking about. If we do that same experiment in a mouse that does not express KMO, we can completely reverse some of those behaviors, which is further evidence that that metabolic pathway that lies downstream of KMO and ultimately results in the generation of quinolinic acid, among others, is likely responsible for driving those particular phenotypes. And one of those is cognitive impairment. And we've measured this now in a few different... Cognitive neuroscience is a really big field. There are many different forms of cognition. And so in a few cases, so we've measured some things like working memory. We have behavioral tasks that we use in the laboratory to get at working memory, or spatial memory, or cue-associated or context-associated learning and memory. And in some of those cases, we can see that this KMO-dependent kynurenine metabolism appears to be responsible for driving the deficits in that particular cognitive function following activation of the immune system.
So it sort of brings up the question of whether these, like behaviorally different things we often think of as different parts of the brain are responsible for them. And the treatments you're talking about affect everything. In fact, even the muscles. So some of the weakness could actually even be muscular, I guess.
Sure.
But inside the brain, you might say, well, there's a place that's responsible for focus and another place that's responsible for fatigue, and that sort of thing. And do we have any notion about that, or even a method for trying to sort that out?
To an extent. Like, we know a little bit about what you're referring to. So we know there are some structures in the brain. For example, the hippocampus is important for spatial learning and memory. It's an important nexus for relaying memories and encoding new information. We know that that's an important physical region of the brain. We know the prefrontal cortex is important for higher level cognitive function, things like executive function, which that's referring to kind of the higher level cognitive processing, complex problem solving, emotional layering on emotional valence and interpretation of subtle and complex, maybe social and environmental things. So we know a little bit about kind of the brain regions that are important for types of cognitive performance, but a little. There's still a lot left to be understood. I mean, you know quite well, brain circuits are complicated. And the interconnectedness within the brain is amazing. While the number of cells in the brain is finite and quantifiable, there's a lot, but it's not completely mind-blowing. The number of potential network configurations and contacts between discrete circuits, synapses, and that layers an order of complexity that can be a little daunting and hard to understand.
Yeah, I say so. So I was just, of course, this symptomology probably, all these different symptoms probably come from effects on different brain circuits, but what is it about an infection that causes, what's changing in this metabolic pathway during an infection that's causing more of these metabolites that are in the quinolytic acid?
So that's a good question. So in understanding, so I guess I'm going to take two approaches to kind of answering or attempting to answer that question. And first is in understanding what's going wrong during pathology, it often requires us to understand the normal healthy condition and how things are supposed to work. And so maybe I should have said this at the outset, is that it's often the case in science when we talk about inflammation, it evokes the notion of a big bad response that is causing all the problems, right? That we want to avoid, we want to stop. And the reality is inflammation is a natural part of our immune response that we need for survival, right? We have to fight these infections. We have to repair damaged tissue and to return to health. The problem becomes when that inflammation is poorly regulated, right? When inflammation becomes too much or it lasts too long. And there are there's a long list of reasons and things that can cause that to become problematic. Now why does that, or how does that change the metabolism? That's a trickier question that we don't fully understand, to be honest. But at least at face value, we know that kynurenine metabolism under normal conditions is a source of NAD biosynthesis, so that it's generating an important cofactor for the cell to perform its functions, whatever cell we're talking about. And when the demands on a cell increase, the energetic requirements increase, it would at least logically hold that the need for NAD increases. It is the case that at least oxidative kynurenine metabolism as a source of NAD, while effective, also can become saturated. So that is to say that the enzymes that are converting one molecule to the next in this metabolic pathway, if you have too much of the substrate, then it can handle and metabolize. Those substrates will eventually be secreted out of the cell in mechanisms that are not entirely clear, but it is the case that, for example, during inflammation, quinolynic acid will accumulate inside the cell, and because it's not able to be metabolized to NAD fast enough, so the cell will start to send it out.
I was thinking about it wrong. I was thinking about making too much, but maybe it's not being able to use up what you've got.
So I think it can be a little bit of both. I think it can be. So especially if we go back into the brain. So if we're thinking about the the kynurenine as the apex substrate, the molecule that then gets broken down to other things. If more of that is coming from the periphery, being transported into the brain, then the brain will metabolize it as whatever the state of the metabolic machinery of the brain is in. So you can have more kynurenine coming into the system, essentially flooding the enzymatic pathways that already exist. And it is also true that those enzymatic pathways change in response to inflammation. So enzyme expression goes up and down. The activity can be modified through post-translational modifications and things. So you can have a little bit of all of the above that results in an overproduction or an accumulation of these metabolites that may have a kind of a collateral damage type of effect. I don't know that the information is completely clear that the induction of the cognitive impairments or the functional changes in the CNS that we were referring about earlier, that these metabolites are evolutionarily designed to cause those changes. I think we have tended to think about them as an unintended byproduct. Like when the immune system becomes poorly regulated, all of a sudden you start to have metabolites build up that aren't supposed to be there. And the system has to respond to the environment that it lives in.
Well, I can tell when I'm getting sick by the change in function of my thinking and my cognition. I think it's super useful. If you ask me, do you just have hay fever or are you coming down with the flu? I can tell you. It's not... The flu does something that the hay fever doesn't do. Okay, so now I'd like to get to maybe the harder or more meaty, speculative question, which is it possible that this particular metabolic pathway gets dysregulated in depression, and that actually depression is a response to an infection that isn't there?
Yeah, so that's a really good question, and there have been a lot of people are thinking about that very question. And while I'm not a psychiatrist, I am a psychoneuroimmunologist. So
good enough for this podcast.
I interact with psychiatrists. And what I can tell you, I think the state of the contemporary science is that we're in an interesting place in terms of trying to understand what causes, say, major depression. We don't know what causes major depression. We know we have a list of drugs that can be prescribed that may help, but that's not to say that the pathways and targets that those drugs hit are necessarily the underlying cause.
Right.
And because depression is, while we used to argue a lot about whether we can study depression in a mouse, and my response to that was always, well, of course not. Because depression necessarily requires a professional, a healthcare provider, to talk to a human patient, ask them how they're feeling, how they're thinking, what their symptom profiles are. And a diagnosis of depression in a medical sense requires a very specific chronicity of symptoms and burden of symptoms. We can't really assess that in a mouse or a rat. But what we can do is measure behavior that a normal animal exhibits and an affected animal exhibits. And we can say that those behaviors may have more or less relevance to some of the symptoms that are experienced by an individual with depression, but not depression per se, but the symptoms may be able to be modeled. And so we think about it in that way, that we're more deconstructing depression into its core features. And those core features may be more easily studied and addressed. But to your question, I think your question that I almost forgot to address or answer is kynurenine contributing to depression?
Yeah. Like, can you just measure levels in the blood?
So you can. And the short answer is it depends.
Of course.
And because depression is a heterogeneous condition, right? So if we have 10 people suffering from depression, they're very likely will have 10 slightly different underlying pathologies and manifestations, yet they all result in the same clinical diagnosis. So I think the prevailing thought of kind of the biological psychiatry community, or at least the people who are thinking about this question, is that there's probably a subset of individuals suffering from depression whose depression is caused by underlying inflammation, metabolic disruptions. And we know, and evidence to support this comes from some of the immunotherapy kind of experiments that happened over the last 20 or 30 years. We know that when human patients who aren't depressed start immunotherapy for things like hepatitis C or certain types of cancer, some of them become depressed when they start their immunotherapy. And there were some clever scientists, some groups around the world, and some in the U.S., several in Europe, that around the early 2000s started to actually study this systematically. And they did see that activation of the immune system in the model system, the immunotherapy models that they were studying, was characterized by changes in kynurenine metabolism. And I'll say those clinical studies were the basis for the first experiments that I started to do in the lab. We were attempting to reverse translate, create an animal model that would reflect what they were seeing in the clinic so we could get at mechanism. Because in clinical studies, especially brain-based clinical studies, you're entirely limited to how far you can go. Like I've heard it once said that the brain is largely inaccessible to science, and we're limited to imaging, clinical, like asking you questions, and possibly collecting cerebrospinal fluid. We have some very limited sampling that we can ethically and reasonably do. In an animal, we don't have the same restrictions, or in model systems, we don't have the same restrictions. I know we have some folks around here who are getting into organoid and cell-based reconstructions and computational things. So there's some really fascinating things going on to try and get at the mechanisms that are underlying those observations that were initially made or have been made in the clinic.
How about modifications of circulating levels of metabolites, and surely somebody's thinking maybe I could treat depression that way. Yeah,
so there's been on two fronts. So there's been some effort to explore using anti-inflammatory drugs as an augmentation or a therapeutic approach to treating depression. There have been some challenges along the way, and I guess to maybe sum up a really somewhat long and complex literature, the evidence that works across the board is pretty poor. Like anti-inflammatories aren't good antidepressants. However, if you restrict those studies to people who have depression and high inflammation, the anti-inflammatories work a lot better to treating their symptomology. The problem is that's not how clinical trials are enrolled. Right. Yeah. So it's been a logistical, a structural limitation in the field for some time now, trying to really be able to get at those questions, because in large part because we don't know what causes depression. The same way with kynurenine. So we've started to think about, the drugs And inhibitors that we have access to use in the laboratory are not available for use in, clinical setting. So they're not approved by the FDA or anything like that. So are there other ways that we can approach this question to either test a hypothesis or try to develop a therapeutic approach, or both? And so we have, and others, but I speak to our efforts, have focused on using kind of nutritional and lifestyle strategies to try and modulate the concentrations of these amino acids or these metabolites that share the transporter that I referred to earlier. So tryptophan, kynurenine, leucine, isoleucine, and valine. These are large neutral amino acids. It's a category of amino acids that are structurally similar, and they all share the same transporter. So as the levels of these individual amino acids or metabolites change, transport across membranes change.
So I could just dilute it in the brain by giving myself a bunch of leucine in my diet.
Actually, that's exactly right.
Uh-huh.
It's been shown in mice multiple times. So my former postdoctoral mentor, who is at MD Anderson now, his lab published that going on 10 years now, giving supplemental injections of leucine is not only able to change how kynurenine flows from the periphery into the brain, but it's also able to mitigate the behavioral responses to LPS. So it's additional, so LPS meaning immune activation.
Yeah.
So additional evidence that is consistent with other findings that we've had, that kynurenine in the brain during inflammation is an important driver of these functional changes. So we recently completed a small pilot clinical trial over at the Barshop Institute for Aging and Longevity. And we were asking the question about whether exercise alone as a lifestyle intervention or exercise plus supplementation with branched chain amino acids, including high-dose leucine, would be beneficial for alleviating symptoms of fatigue and cognitive impairment reported by older individuals who report high baseline fatigue. And while it's preliminary, the results are actually quite promising. They showed a 42% improvement in fatigue symptoms that are reported by the individuals taking the amino acid supplementation plus exercise as compared to exercise alone. So it's not, again, it's a pilot study. There are some important control groups that we weren't able to complete because of limitations in time and resources. But at least it's a basis to support asking the question on a bigger scale and seeing, can we do something to help improve people's symptoms by modulating this metabolic pathway under certain conditions. So these are things we're thinking about
That's
currently.
just great. I mean, what a good way. If I had to pick, how am I going to be treated for some disorder? And taking a bunch of amino acids and running sounds like that's what I would pick. What a good way to do it.
Yeah, I mean, if it plays out that way, I mean, that's ultimately the science will tell. Yeah. Right. That's why we have to do the experiments, and that's why we have to generate the data. But I'm right there with you. I think if for using exercise as an example, we all know that exercise is good for us, but few of us actually partake, at least consistently. That's especially problematic as you start to think about older individuals who perhaps have physical limitations, other medical comorbidities, things that make starting and maintaining an exercise program even more difficult. If, in addition of an over-the-counter dietary supplement plan, is able to reduce the threshold of exercise needed to achieve a benefit, or allows them to feel better faster. Because I think that is oftentimes one of the, and we're bouncing around a little bit, but when we think about exercise, it hurts if you start an exercise program de novo, like it hurts. You're sore, you're uncomfortable, and if you don't start to feel better from it, it becomes really hard to maintain. Just on the basis of intellectually, I know that it's good for me. So perhaps that's something that, we could move the needle on that. There may be other elements in play from a biological basis that, we're kind of approaching the questions from multiple angles. And we don't have time to talk about all of them today, unfortunately, but these are the questions that we're thinking about. Ultimately, when I became a scientist, when I went to graduate school and said, I want to be a biomedical researcher, I'm doing experiments in the lab using cell culture and animal models, and ultimately it was my hope that something that I did someday made a difference in someone's life in a positive manner. And so I feel like we're kind of at a place in our research program that we're starting to transition into that realm to say, do the things we've learned in the lab apply in the real world in people? And at the end of the day, like I said, that the science will tell if it does or not, but that's where I want to be. I think that's what's exciting.
That's just great. Good luck with that. I hope it works out perfectly well.
Well, thank you. I hope so too.
Thanks a lot for joining us.
Yeah, this has been fun.
This has been Neuroscientists Talk Shop.